Hello, my name is Dave Rice, and I'm the Technical Director of the NMR facility. The NMR facility is a shared instrumentation facility for the UC Merced campus, which is designed to primarily provide nuclear magnetic resonance. But we also provide a bunch of other techniques, including LCMS. And a colleague of mine, Murad Sadki, is going to talk about LCMS shortly. NMR is primarily used for molecular characterization, or to characterize molecules. All matter is made up of molecules, which are made up of atoms, and the purpose of NMR is to figure out the structure of the molecules. Our users at UC Merced are primarily chemists. Chemists study matter and molecules, and also biochemists who study biological molecules, and engineers who also do chemistry and biochemistry. These days, molecules can be anywhere from very small molecules. For example, pharmaceuticals or drugs are examples of small molecules, and the chemists at UC Merced make these molecules, and then they bring them into the NMR lab for structure determination. But some of the molecules we look at are very, very large. They're biological molecules, and NMR can also be used to determine the structure of those. The third part of the NMR experiment is data processing. And on this computer screen is the typical result, an NMR spectrum. And I mentioned that the NMR spectrum detects the protons in molecules, and each proton has an individual peak. And the chemists can interpret the peaks to determine the structure. So we have two NMRs in the basement here. This is called a 500, and behind me, which you'll see in a moment, is a 400. And NMR is made up of two parts. The first part is a very high field magnet, which is this white can in front of you. The second part is the electronics to excite the molecules. And that's all in this chassis sitting right here. NMR actually detects the hydrogen nuclei in molecules. And from that detection, chemists can understand the structure of molecules. So this is a typical NMR sample. Chemists typically place their molecules inside a dissolved in solvent inside of an NMR tube. And then they place the NMR tube inside the magnet of the NMR spectrometer. So we have a second NMR called a 400, which is used for the same purposes as the 500. So this is the NMR facility. And so next, we're going to head next door to the mass spectrometry facility.
Hi, I'm Murad Sadki. I'm a project scientist and the director of the mass spectrometry facility for CCBM, the Center for Cellular and Biomolecular Machines at UC Merced. The center was established in 2016 with $5 million grant from NSF, the National Science Foundation. We are located at the basement of the Science and Engineering Building, too. Before starting the virtual tour, let me explain what is the mass spectrometry. Mass spectrometry is an analytical technique that measures the mass-to-charge ratio of particles in solution, which is a way that we can separate atoms, isotopes, and even fragments of molecules based on their mass. Let's look at how mass spectrometry works. This is a hybrid instrument from Thermo Scientific called Q-Exactive. This is showing the schematic design of the instrument with front inserts and S-lens and bent flat topor for cleaning ions, quadrupole mass filter for ion selection with an HCD collision cells and an Orbitrap in the back for detection. In full scan mode, the ions are produced at the source and captured and focused for effective ion transmission using a variety of lenses. Uncharged molecule species at this point are filtered out using the bent flap topor, and this is a full scan MS analysis. All the ions are passed through the quadrupole, collected into packed in the C-trap and stabilized and sent to the Orbitrap for detection. In the Orbitrap, the ions are trapped in an orbital motion where the frequency of rotation is related to the mass-charge ratio of the ions. for ions with different mass-charge ratio will have different frequency of oscillation. Presently, the mass spectrometry facility houses two advanced mass spectrometer instruments from Thermo Scientific, with the ability to analyze samples with high resolution and mass accuracy. Here we have a Q-Exact hybrid quadrupole Orbitrap coupled with the ultra-high performance liquid chromatography Vanquish system. This instrument is part of the Center CCBM and is mainly used for biomolecular samples, peptides, large proteins, and oligonucleotides. Here we have the Exactive Plus, which is a hybrid Orbitrap mass spectrometer coupled with the ultra-high performance liquid chromatography Ultimate 3000. This instrument is part of the School of Natural Sciences and was funded by the Department of Defense. The instrument mainly used for small organic compounds. Here are some of the research projects we are working at UC Merced that the mass spectrometry facility supports. In particular, we are developing protein-based biosensors at the cellular level, which are able to probe different properties in the cell like pH and different concentration of molecules. These biosensors have potential biomedical diagnostic application, and currently one of the projects we are working on is to make a biosensor-based diagnosis of COVID-19. So mass spectrometry is an essential analytical tool in the initial phase of many research projects to assess the purity and identity of the biomolecules and small organic compounds. Let me show you in real time how a mass spectrometry works. Here I have a recombinant protein which was expressed and purified in the lab, and we want to assess the purity and the identity of this molecule. So the sample, we dissolve the sample in an organic buffer compatible with the mass spectrometry, and then will be injected in the syringe by a direct infusion. And the solution, the liquid solution, will be converted to the gaseous phase in the ion source. And then the ion will go through the quadrupole and then get detected by the Orbitrap at the back of the instrument. Here are the spectra of this. This is the spectra of this molecule, which we have the mass to charge ratio in function of the relative abundance. And then we can convert this spectra by deconvolution, and we can calculate the mass spectrometry, the molecular weight of this molecule. And then we can compare the molecular weight to the theoretical molecular weight by looking at the sequence of the amino acid. And we can see this protein, the molecular weight of this protein is 8062 Dalton, and compared with the experiment we just did, we have the same exact molecular weight, 8062 Dalton. We can assess now that this protein is pure and there is no other complexity or impurity in the sample. Our facility provides the opportunity to be a hub of innovation at UC Merced, connecting scientists with the tool and expertise that can take their research project to the next level. We look forward to work with you.